Hello viewers, this is Dr. P. Mary Anupama. Thank you for watching Anupuma's Biochemistry, and here I am with another topic, Introduction to Microbiology. What is microbiology? Microbiology is the study of organisms which we cannot see with the naked eye. So these are too small and we have to observe them using microscopes. These include wide varieties of organisms that include bacteria, archaea, fungi, actinomycetes, algae, protozoa, viruses, and so on and so forth. Microorganisms include first bacteria and archaea. These are prokaryotes and they have no organized nucleus. While we have the other category which come under this fungi, actinomycetes, algae, protozoa and nematodes. All these are eukaryotes and they have organized nucleus. So here this is the archaea which has an unorganized nucleus, while this is the bacterium which doesn't have an organized nucleus. Just the nucleoid is directly present, the DNA is present at the nucleoid. So in the case of eukaryotes, they have this organized nucleus, and we find all these things in air, water, soil, inside and outside the animal bodies, even our human beings, we find these things. So what is the importance of these microorganisms? They operate all these cycles, that is the carbon, nitrogen, phosphorus and sulfur cycles. Some important products we obtain from these microorganisms. These include bread, cheese, yogurt, alcohol, wine, beer, enzymes, organic acids, antibiotics, vaccines, vitamins, and so on and so forth. Today we even have this genetic engineering techniques. These led to what are known as the recombinant bacteria, and these are used to produce many important proteins which are of useful to us, like the insulin, hormones. We have recombinant vaccines, then interferons, biopesticides, even bioremediation is because of the recombinant bacteria, and biofuels. Let us see the golden age of microbiology. Here is an experiment of Reddy experiments we call it. So here he was talking about the spontaneous generation of the microorganisms. When decaying meat was just kept isolated from flies, maggot could never develop. So this is the sealed flask. When meat is exposed to the flies, then immediately the microbes they erupted out. And then here this is the flask covered with gauze. Again, no, nothing is seen. So as a result, scientists they started doubting about this Aristotle theory, which is about the spontaneous generation of microorganisms, and Reddy's experiments has proved that there is nothing like spontaneous generation, because maggot carried some, because the flies carried something over there, the maggots appeared. When it is totally sealed, nothing has appeared. So they tried to disprove the Aristotle theory of spontaneous generation. One more experiment which we can see is the Pastrious experiment. He has used this swan-necked flask, and these were kept upright. So when they were like that, there was no microbial generation. Here you can see they took the medium and they infused it. So the steam, it just escaped out, and as a result the air which is present inside it is removed. Now whatever, they just left it like that, and whatever dust is entering, it has settled here just like that. And here what they have done is, now as it is upright, the dust has settled here. They tilted the flask such that the medium comes into contact with the dust. So the dust has microorganisms. So the microorganisms they started growing in the sterile infusion medium, and microbes started appearing. So when the flask was tilted, the neck was bent and the dust has went inside, and the infusion medium became cloudy because of the growth of microorganisms. Next, slowly they started tracking out the various microorganisms and their applications. So various microorganisms they identified. This is some hundreds of years ago in the 18th century itself. They found out the microorganisms uh, in the cheese. They are being used for flavoring and ripening. Okay, both bacteria as well as fungi were used. And flavors they depended upon the source of the milk and the type of microorganisms that they are using. Next, alcoholic beverages were produced by fermenting the sugars or fruit juices with or the grains with yeast. Then soya sauce, this was produced by fermenting soya beans with fungi. Vinegar was produced by bacterial fermentation of sugars. Yogurt, they took skim milk and they added bacteria and they got yogurt. Then sour cream was obtained when bacteria were grown in cream. Then artificial sweeteners like amino acids synthesized by bacteria and sugar, they are also available for us. And bread, this is again dating back to 18th and late 18th centuries, and all 18th century they had lots of bread. This is produced by the raising of the dough by the action of the yeast. So soaring dough, it resulted in bacteria to produce acids, and then that dove, that puffy nature of the dove was because of the release of carbon dioxide by the yeast. So all these industrial use of microorganisms, they started coming out during the late industrial era, that is sorry, during the late golden age of microbiology. And some more industrial use of microorganisms that we can see is the production of these antibiotics and all. Both bacteria as well as fungi produce them. Today you even have this genetic engineered bacteria that produce growth hormones and insulin. Laundry enzymes like the protease, amylases, they are produced by bacteria. Vitamins are produced by bacteria. Then we have this diatomaceous earth or the kieselguhr, which is used in polishes and buffing compounds. That comes from the diatoms, that is the microscopic algae. From their cell wall you get it. Then you have this pesticides, that is biopesticides, which are produced from bacteria. Then drain openers, that is protein digesting agents, fat digesting agents, that is the enzymes produced by bacteria, they are being used as drain openers. Disease causing agents were also identified in the good olden days, where Pasteur he developed this germ theory of diseases, and Robert Koch he studied on the causative agents like this anthrax, and he even examined colonies of microorganisms and studied them very well. Koch's experiments were the major contributors. He was the person who has contributed a lot, like the simple staining techniques. The first photomicrograph of bacteria was by Koch. Then bacteria in the diseased tissue, that photomicrograph was also by Koch. Then the techniques to find out the colony forming units, how to estimate the colony forming units per ml of water or per ml of medium was by Koch. Then the use of steam to sterilize media was by Koch. Use of petri dishes, antiseptic techniques, bacteria as distinct species, that was also Koch's thing. And then Koch postulates. All these are the contributions of Koch to the field of microbiology. So many notable scientists they have contributed to this. We see that anthrax by Robert Koch, and Neisser about gonorrhea, Charles Leveran malaria, like this again Koch tuberculosis. Then we have Albert Frankel pneumonia, again Robert Koch cholera, David Bruce undulant fever. Like this, you can see lots of major contributors to this field of microbiology. Even the disease prevention, prevention of infections, that was also very well studied. Semmelweis, he was talking about hand wash. Lister, he has come up with antiseptic techniques. Nightingale about this nursing. Then Snow, he was talking about this infection control and epidemiology. We all know about Jenner's vaccine. He has contributed a lot to the field of immunology and vaccination. Ehrlich, he was talking about this magic bullets in the field of chemotherapy. So with time, as they have studied that it's microbiology is not a small thing, microorganisms are not just one. There is a lot which they can contribute to. So various more disciplines have come up in the microbiology, and the scientists who contributed to this include this Leeuwenhoek. He was a major contributor to bacteriology, protozoology, mycology, parasitology, and phycology. Linnaeus to the field of taxonomy, then Semmelweis and Snow to infection control and epidemiology. Then we have Pasteur. He was talking about pasteurization. From here, it's about the industrial microbiology which has started. He contributed a lot to food and beverage technology. Then Buchner, he was working on microbial metabolism, genetics, genetic engineering. Koch postulates it was about etiology. Ivanovsky about virology. Bejernik and Vinogradsky about environmental microbiology. Again, that's a unique thing. Ecological microbiology was there. Then Gram was contributing to microbial morphology. Today we hear this gram positive and gram negative bacteria. Lister and Nightingale, they were talking about this antiseptic medical techniques. They have come up with that, and even hospital microbiology has evolved. Jenner, von Behring, Kitasato, they contributed to serology and immunology. Ehrlich about chemotherapy, and Fleming, the pharmaceutical microbiology. The major disciplines of microbiology, which all of us know, are this bacteriology, which is about the study of bacteria and archaea, phycology about algae, mycology fungi, protozoology about protozoans, parasitology, it's about parasitic protozoa and parasitic animals, virology about viruses. So these are microbe centered. Now we also have this process centered ones, wherein you have this microbial metabolism. It is again a separate field of biochemistry where chemical reactions taking place within the microbial cells is studied. Then we have this microbial genetics, which talks about the functions of DNA and RNA. And we have a unique discipline of environmental microbiology, which talks about the relationships between microbes and various microbes, that is interaction between the microbes and the other organisms, as well as between microbes and the environment. So under medical microbiology, okay, all this is about applied microbiology. We have medical microbiology, applied environmental microbiology, and industrial microbiology. Medical microbiology includes serology, which talks about antibodies in blood serum and again indicators of infections. Immunology is about the defense system. Epidemiology is about the frequency of a disease, the distribution of a disease, or how it is spreading. Etiology is about the causes for a particular disease. Infection control, it talks about the hygiene and healthcare settings. Okay, it talks about the control of nosocomial infections. Then we have chemotherapy, which talks about development and use of drugs to treat infectious diseases. Applied and environmental microbiology includes bioremediations, that is use of microbes in pollution control. Then public health microbiology, wherein we can use them for sewage treatment, water purification and all. Agriculture microbiology is about use of microorganisms in agriculture as biopesticides or biofertilizers too. Industrial microbiology, it includes the food and beverage industry, wherein you can reduce or eliminate harmful microorganisms over there. Pharmaceutical microbiology is about productions of antibiotics or vaccines and all. Recombinant DNA technology, it's about producing useful products to us or manipulating with the microbial genes and getting some vaccines and all such things. So here you can see some basic important things about biochemistry. What are the basic chemical reactions in life? Like it began with the Pastrious work on fermentations, and Buchner, he has discovered the enzymes in yeast cells. Then Kluwer and Van Niel, they identified that microbes, they have used microbes as the modern systems for biochemical reactions, and many practical applications have come up, that is design of herbicides, pesticides, and even the diagnosis of illnesses and monitoring of patient responses to treatment of metabolic diseases, then drug design. All these are the contributions in the modern age of Microbiology. So today we also talk about how do genes work in the microbial genetics. We have molecular biology, which has evolved recombinant DNA technology, and even talk about gene therapy using microorganisms. When it comes to recombinant DNA technology, the genes which are present in the plants or microbes or animals, they are manipulated, so that we can use them for some practical purposes. Like we are able to produce human blood clotting factors using E. coli, and they are using them to treat hemophiliacs in those people where the blood coagulation time is more, or they'll be bleeding if they get any cuts or bruises. So if you can introduce these blood clotting factors and give them a relief. Next is gene therapy. See, we have genetic defects. Some people may be lacking some genes, some genes may be missing or some genes may be defective. So you can just simply introduce that defective gene and then get the host back to normal functioning. So here, say for example, here the cells of the patient, they are removed, okay, and then they're taken to the laboratory where the viruses having your gene of interest, they are infected to these cells, and as these gene gets inserted, these are the manipulated viruses. As these viruses attack the cells, random recombination will be taking place, and you have the cells, the human cells, back with uh, genetically altered one. That means the defective gene is replaced by a functional gene, and you can replace it back into the individual. So now the individual will be functioning normal. That means the defects which you find because of loss of gene, it is repaired. It is repaired. So this is what is gene therapy about. The role of microorganisms in environment, already we talked about bioremediations using bacteria, fungi and algae. We can do it, and recycling of chemicals can be done, that is carbon, nitrogen and sulfur, that cycles is because of microbes. And in the modern age, we have this serology, which I have been already telling about. It's the study of blood serum, the antibodies and all. So Von Behring and Kitasato, they talked about the existence of some chemicals in the blood and the cells that the cells can fight, and all such things were studied. And then we have immunology, wherein you talk about defense system, chemotherapy. Here we have this penicillin discovery by Fleming, and even sulfur drugs discovery by Domack. So these two are the major contributions for chemotherapy. Some more important revolutions you can see here, finding out the zones of inhibition. So bacteria, like what we do is, this is the pore plate method, wherein into the nutrient agar, which is solidified and autoclaved, when it is reaching around 37 degrees centigrade, you add little bit of microbial culture and pour into the petri plate. Then you take a fungi which can produce some antibiotics. Say for example, it can produce penicillin. So add a drop of that fungus, just one or two, dilute it in such a way that you just add a drop of it at the center and leave it like that. So when this is producing, say for example, penicillin, you will find the zones of inhibition. So here the bacteria will not be able to grow because it is sensitive to penicillin. So like that, you have here streptomycin colonies, they are producing blue antibiotic, which is studied. Thank you for watching this video.